Okay, so hi everyone. My name is Vassi, as Patricia said, and I'm going to talk to you today about the icy moons, exploring the icy moons in our solar system, and could life be out there? So this is just going to be a bit of an overview over some of the icy moons in our outer solar system. I'm not going to go through all of them, I'm sorry. um, as well as some of the chemistry that is of interest to us. Why are we drawn to these certain moons? Why are they interesting to us? And what could we possibly find out from looking at them even further? So to start off with, let's clarify what we mean by life when we talk about could life be out there. We do not mean aliens as you expect them in your mind with sort of like, the big head, the green or gray head, or necessarily even high intelligent life. We're not necessarily talking about that. Life could be anything from microorganisms to microbes, like strain 121 here on the left, in the picture on the left, or even down to small animals, like you can see the tardigrades or water bears, as they're known, on the right-hand side. A fun fact about water bears and tardigrades, actually, one of my collaborators and close friends, Alejandra Traspas, published a paper on them this year, where she looked at their ability to survive impact shock at very high speeds and pressures. It was actually one of the most read articles of 2021. So there's a lot of interest in these kind of things and all sorts of microbes in life. And could they traverse space and the whole theory of panspermia? And what she found actually is that they can survive impacts up to about 0.9 kilometers a second under vacuum, which is incredibly impressive. And here on the bottom, there's a picture from the microscope that she took of her tardigrades. And I have the little reference there if anyone wants to go look at that paper. But it was very popular. It had a lot of coverage. So I thought some of you might have come across it. I thought I would include it here for you. But this is essentially what we mean by life. We don't necessarily mean a whole big structure, but it could be something very small, tiny, down to single cells. At the moment, we haven't really found very much anything exciting really outside our planet. We have a lot of interesting things, sure, but life as we know it, not quite there yet. So before we get into it, let me just tell you a little bit about me. So I initially had my first degree in forensic science and then went on to do my PhD in chemical ballistics. So I came from quite a chemistry, forensic, physicsy background. And then a few years ago, when I finished my PhD, I started a job as a postdoctoral research associate, which is just a fancy way of saying a researcher, in astrochemistry and space science at the University of Kent within the physics and astronomy group. And what I have been focusing on these last few years, what the main focus of my research has been looking at the shock synthesis of prebiotic molecules in the solar system. So essentially, very simple prebiotic molecules such as amino acids. Can we synthesize these amino acids in the universe just through the hypervelocity impacts that occur all the time from comets or meteors, meteorites, all sorts of things? Could the force of those impacts, is it sufficient to create amino acids if the ingredients for life are out there? So that's kind of what I've been focusing on, and I've had a special focus on the icy moons. So I'm going to give you a little bit of an introduction into the icy moons and talk to you about a couple of the icy moons and why they're interesting. And then I'll bring my research into it and tell you a little bit about how that relates to them. So like I was just saying, a quick overview. So I'm just going to give you a brief introduction and some background on the icy moons, just to provide a bit of context and show you why we find them interesting. And then I'll go into about whether there could be life in the outer solar system, as well as the introduction to my research and how that relates to the origin of life in our solar system. And then we'll go on to continue about what future work or plans there are in motion with regards to this field. So just a brief introduction. Many of the outer moons are suggested as places where extraterrestrial life could exist. And specifically with the ones that we're going to focus on is on the far left, top left, Europa, and the top far right, Enceladus. Those are the two icy moons that I'll be talking to you about. And the reason that these two are interesting is due to the presence of subsurface oceans. Although our planet is very abundant in water, if you look around at our near neighbors, water is quite scarce. You don't see big pools of water on Mars. They're said to have evaporated long ago, and the same on Venus. But out of all the rocky planets, Earth has a very high abundance. And in the past, we thought that the icy moons were sterile lumps of ice and rock, nothing more to them really. However, we recently, and I'll go into a bit more, we have found that actually it might not be as clear-cut as we think. So to start off with Europa, firstly, Europa is one of Jupiter's icy moons. It has a surface temperature of about minus 130 degrees Celsius to minus 220 degrees Celsius towards the poles. And it has a crust of ice on the outside. So on the left-hand side in the picture that you can see the whole ice crust or shell, if you like, and you can see some of the striations or lineae, as we call them, which shows you some of the cracks or weaknesses in the ice. And on the right-hand side, we can see the model that shows you our current understanding of Europa. So what's interesting about Europa and why we're so captivated by the possibility that life could exist on this moon is because, as you can see on the diagram, on the right-hand side diagram, bottom left corner, where you can see the little moon of Europa, you can see that it has an iron core that is surrounded, sorry, mosquitoes, that's surrounded by a rocky mantle. And on top of that, there's a saltwater ocean beneath that icy crust. So the fact that there's an ocean in there, and it's not just ice, is already making people think, well, on Earth, we have the theory that where there's water, there's life. Therefore, if there's water in there, could that possibly mean that there is life? Similarly, if we look at Enceladus, so Enceladus is one of Saturn's moons, another gas giant. So the moons of the gas giants are the ones that are of high interest to us. So Enceladus itself is very white and bright due to the icy surface, and is actually the most reflective body in the solar system, with a surface temperature of about minus 200 degrees Celsius. So very, 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 very cold. However, thanks to NASA's Cassini spacecraft, we managed to observe geysers on the South Pole of Enceladus. And you can see here on the top left-hand side, the geysers that are coming down they're coming out from the southern pole of Enceladus. And underneath, I have a picture for the Baghdad sulcus, which is essentially the tiger straps where these jets of water vapor and silicates are all coming out from. And what we find is that actually the material, again, thanks to Cassini, is actually being ejected at about 400 meters per second. So, and for that to be ejecting for that long and consistently, it suggests the presence of a subsurface ocean, sorry. Now, we also believe that Enceladus could have the presence of hydrothermal vents. As you can see on the top right-hand side image there, you can see the hydrothermal vents just over its core on that inner rocky area, releasing material into the water, but then ultimately some of it comes out from the southern poles of Enceladus. And the reason we think that Enceladus could have hydrothermal vents is because small bits of silica have been detected in those plumes through observations that can be generated when liquid water and rocks interact at temperatures at above 90 degrees Celsius. So hydrothermal vents have the ability to produce quite high temperatures because they are so close to the Earth's core. And the environment there is only really hospitable to what we would call extremophiles, things that like extreme temperatures. And However, these subsurface oceans and the possibility of hydrothermal vents have made Europa and Enceladus, amongst other icy moons, I should emphasize, to have significant astrobiological potential. But why are the hydrothermal vents interesting specifically? Well, I'm glad you asked. If you recall from one of my earlier slides, strain 121, which was a microbe that actually exists in hydrothermal vents on Earth. So deep at the bottom of the ocean, at temperatures of minus 121 degrees Celsius, which is where it gets its strain name from, these microbes happily exist. Hydrothermal vents and black smokers can reach temperatures down to almost 300 degrees Celsius. But we have found these at 121 degrees Celsius, which is above boiling. So it's like water boiling temperatures, which is pretty impressive. So if we use Earth as a model for that, and let's be honest, it's the only real working model we have that we can observe, it could suggest, the data we have could suggest that there could be life near the hydrothermal vents or in the oceans of Enceladus or Europa. However, if that was the case, life could be quite different. It could be more aquatic in nature. It could be much more simpler than we realize. It wouldn't need light. It wouldn't need, like here on Earth, a lot of us are creatures of light. Plants need light, we need light. It would be a completely different biodome that we would have to investigate. But again, that's assuming that the theory we have based on Earth's model, that where there's water, there's life, that is actually true. So here is where I'll bring in a bit of my research. So like I was saying, on Earth, we have this theory that where there's water, there's life, which is true as far as we can tell, but only to a certain extent, as in order to actually start more complex life, or like organisms in life as we know it, we probably need to have some sort of complex organic precursors like amino acids to be present and somehow to be made. So amino acids are known as the building blocks of life because they are one of the most primary structures, organic structures you can get, that then can go on to make longer chains and branch out to make proteins, which would in turn make DNA, and then the possibilities are endless. So what we've seen is that water ice and carbon dioxide ice are present throughout our solar system. Then through observations of various bodies, we have found ammonia ice on the surface of Triton and Jovian clouds, Saturn's rings, and Enceladus, as well as carbon, nitrogen, and nitrogen-bearing compounds in spectra from Europa, Enceladus, Ganymede, Callisto, so many different bodies. And the reason I listed these elements here is so you can see on the right-hand side, I have just an average amino acid. So you have your main sort of group in the middle, however long your chain is. And on the left-hand side, the part that's in a red square, that's the amino group. That's what makes it the amino acid. So as you can see, having carbon dioxide or carbon nitrogen could imply that you can make these structures if we know that they exist. Water ice, the water would bring hydrogen and could bring the oxygen, as well as the CO2 ice, of course, to create these structures. Ammonia as well could bring nitrogen and hydrogens along with them. So those are things that basically you're thinking, well, the ingredients of life are all out there. Doesn't mean they can necessarily come together. We will have a look at that in a little bit, but it means that we have them there. So essentially, my research has been focused around whether we can actually make these sorts of amino acids through impacts. If we have the ingredients out there, if we impact them with something that can produce a sufficient energy, would that in turn create that synthetic reaction to bring these together? Essentially, to put it plainly, if you have, let's say, eggs and milk and flour and you hit them all together, are you going to make a batter, Or are you just going to have broken eggs, spilt milk, and just flour all over the place? So that's one of the things I've been looking at, and I've been focusing specifically on impacts. Although there are many phenomena that are experienced by bodies in space, like irradiation and all sorts of things like that, I specifically have chosen to focus just on impacts, hypervelocity impacts that could be caused by asteroids or meteors or comets, any sort of impact when anything really hits anything. And why impacts, you might ask? Well, first of all, impacts are very common in our solar system. When you look at the solar system's history, it might not be every day here, but they are fairly common and they are ubiquitous throughout our solar system. They have been responsible for a range of constructive and destructive events from the solar system's early history and can cause very high shock pressures and temperatures. And these are very important if you're trying to make things, because these are the reactions that produce that energy that you will require to see if you're going to bond things, if you're going to make things, if you're going to mix them and create something new out of them. They can occur at very high velocities, and a significant advantage of them is that we can emulate them using our experimental facilities. So here on the right-hand side, I have some pictures of the craters on Europa to show you how massive they can be. And even Enceladus has craters up to about 35 kilometers in diameter. So a lot of these impacts are huge, and we don't really realize the size of them. There's a scale bar there on this picture. It's 20 kilometers across. So yeah, they can be huge, and they can cause so many things as a result, so much destruction, but can also end up being constructive. So when I said about emulating impacts using the Kent light gas gun, here's a picture of it. I felt like I had to show you it, firstly because it's a key part of my research, and secondly so that you realize it's really underwhelming. Because when people hear a two-stage light gas gun, they think, wow, but in reality it's just bits of metal welded to more bits of metal. But all these bits of metal actually have the ability to let us fire any sort of projectile, well, within a certain remit, but a variety of projectiles at speeds ranging from about 200 meters per second all the way up to 7.6 kilometers a second, all in the lab under vacuum. But we can vary the pressure if we don't want it back under vacuum. Yes, it's very useful to actually try and emulate solar system impacts in the lab. So looking at the picture on the right-hand side, I have the schematic there for you, but on the right-hand side, the green side closest to us is the pump tube side, and that's where you would put in your cartridge. And then further forward, if you see on the diagram, it says the sabot, that's where you would put in your projectile. And that would get fired down towards the big box right at the back, the big white box in that image, as well as in the schematic, which is the target chamber. And that's where your target would normally go. So a bit of background as to how my project came to fruition and where these ideas all came from. So glycine has been detected on several different bodies, including Comet 67P. It was found, do you see the little atmosphere around it? It was detected around there as well. So glycine is an alpha amino acid. So it's one of the basic amino acids. And we thought, hang on, well, how could that have got there? But then back in 2010, Goldman and his team carried out some computational simulations where basically they shock synthesized material. They had a mixture of ammonium hydroxide, carbon dioxide ice, and water ice, I believe, if I'm not mistaken. And essentially they compressed it using the computer. Simulation just compressed it at about 9 kilometers a second, as if it was experiencing a 9 kilometers per second impact. And they found that once they let it go upon condensation, a lot of these had bonded together to actually make amino acids, and glycine was one of the amino acids that happened. So based on this information, back in 2013, Zita Martins and my former boss at Mark Price thought, okay, this is actually very interesting. Let's see if we can replicate these results experimentally, because it's one thing to do a simulation on a computer, but reality could be something very, very different. So they made the exact same type of mixture, of ice mixture, and they impacted it, and they found that between 7 and 7.15 kilometers a second, so within quite a very small margin, they found that they did actually manage to synthesize glycine, and that was just through the force of the impact, which was quite interesting. So here in the middle, I have the chromatograms from their work, and you can see on the top one is the control sample, which doesn't really, it just shows you the blank. And then underneath is the actual target sample, and you can see very clearly circled in red the 3 peaks that showed up that did not exist in the control sample. And these were ran at the same time. The only difference was the sample got impacted, but the control didn't. It was still in the chamber and experienced the exact same conditions. It just didn't get hit by a projectile, so didn't experience that compression, that high energy. So you can clearly see that they actually managed to synthesize that. Therefore, not only did they confirm Near Goldman's simulations, but they actually confirmed to everyone that impacts could cause amino acids. And therefore, the presence of glycine on common 67P could possibly have originated through impacts. Like I said earlier, a lot of the material is already out there in separate forms, so not all together as an amino acid. But if you have a carbon dioxide ice, water ice, some ammonia, and I guess the right conditions are very important, then you could synthesize glycine through impacts. That is not to say that it didn't get synthesized a different way, maybe through irradiation or something like that, but it gives you a very likely possibility as to how it could have happened, or at least an option as to how it could have happened. So then following on from that, last year, we did some experiments where we wanted to say, okay, so now we have glycine. We're taking the observations and we're taking all the prior work and saying, okay, I believe you, we have glycine. What would happen then if you have glycine and it gets impacted again? So if on that cometary body, if that glycine present on there was to be impacted again by another comet or asteroid or anything, what would happen to that? Would anything happen to that? Would it break down the glycine? What would happen? To try and basically understand the survivability of this. Is this something that if it's synthesized one, it kind of just stays there? Or was it a fluke that it was made in the first place? And what we found is that when we impacted an ice mixture of glycine, so water ice and glycine, at 7.6 kilometers a second, we actually ended up forming isoleucine, which essentially adds to the chain. So as you can see here on the right-hand side, I have the structure for both glycine and isoleucine. And you can see that they're very, very similar. Isoleucine is just slightly longer, has a few more hydrocarbons added to it, which is interesting because it shows you that actually, who knows how far this could go. But it's worth noting, obviously, that the conditions have been very, very different. The first simulation showed you about 9 kilometers a second. The first experiments that they did in 2013 showed you a very narrow margin of 7 to 7.15 kilometers a second. And then the work that we did shows you 7.5, 7.6 kilometers a second. So there's a high amount of variation when it comes to this, which makes you think a bit more about the conditions being needed. That just because the materials are there, it doesn't mean the conditions are always going to be right. And that slight changes in these conditions could cause very big differences. So, but then once you have glycine, as you can see, we started making longer hydrocarbon chains. But then how can you start making a bit more complex, how can you start making a bit more complex compounds from that? Well, this is where cysteine comes in. This is another primary alpha amino acid. However, it has sulfur in it. So as you can see, its structure is very similar to glycine that we just saw, but it has a sulfur on the end. And the sulfur is very interesting because it actually has the ability to form disulfide bonds. As you can see here on the right-hand side, the two sulfurs can start causing branches, and those branches can lead to tertiary and 3D structures and create proteins and more complex molecules. So it would be very useful if we could detect cysteine, because then we would know that we're on that next step of branching. The interesting thing about sulfur is that it's actually present on the surface of several Galilean moons, including Europa. Now, this is very interesting because, like I told you earlier, Europa already has a lot of these available, and the sulfur is sort of an addition. So it's not innate to Europa. Well, that's a bit debatable because there's some debate as to whether there is sulfur in the ocean, but we won't touch that because it's still under discussion, and how much it would affect sort of the top layer, the surface that we're looking at, is questionable. But we do know that sulfur originates from Io, the volcanic moon of Jupiter, and actually gets transported all the way to Europa, after which it then gets embedded in Europa's ice. So if you have carbon and nitrogen present, and you have the saltwater ocean, subsurface ocean, and ice, if you have all that, and including sulfur, you're thinking, okay, well, we have all the materials. If we impact it, would the energy be sufficient for cysteine to be created? And this is one of the things that we have been working on at the moment. We're still a bit behind because of COVID and lockdowns and everything shutting down. And you know how things are, especially if you are an experimentalist like myself. If you can't work from home and you have to be in a lab to get data and perform experiments, things move slow when you're not allowed. But at the same time, it's an interesting premise because it's showing you that the ingredients are there. And if we can show something like cysteine be created, you're showing the different steps. You start from something like a base, you're starting from the ingredients. You then go a step above and you manage to make primary amino acids like glycine, just from impacts, without anything else, just impacts and the materials that already exist. You impact that further, you make longer hydrocarbon chains. Okay, that's interesting. If you add sulfur, could you make that? If you make cysteine, then does that mean you can start making tertiary structures? You can start making more complex proteins. And then the possibilities really are endless because there's so many materials that you can make from that the second you can start branching. So that's one of the things, like I said, that we were looking at. And this is how we do it in the lab. So ignore the little plastic things and the sensors coming out. That was to measure something in the ice. But essentially what we do is we make an ice target on the left-hand side, as you can see. And the ice target will have a similar composition to whatever it is that we're looking for, whether it's a similar composition for the surface of Europa, or whether it's just a saltwater ice target, or whether it's just a glycine ice target. Depending on what we're looking at, we modify it accordingly. But we make that ice mixture, and then we freeze it in a carbon dioxide freezer, which takes it down to approximately minus 120 degrees Celsius. And once it's thoroughly frozen, we leave it there for a couple of days. We then take it out, put it in the gun, put it under a lower pressure. We can't go under vacuum, unfortunately, with ice shots, just because all the ice would sublime away. But we go to sort of about 500 millibar and impact it. And on the right-hand side, you see what it's like post-impact. Again, ignore the little plastic rods. They're just holding up a sensor. But basically, this is what we have at the end. And then we take this target and we slowly evaporate the ice to make sure we don't lose anything but the water. So we evaporate it very slowly over a span of a few days to take away all the water and leave behind any residues. And then we take those residues and perform a whole bunch of analytical chemistry steps on it. And then we run it through a gas chromatography mass spectrometry machine in order to get the identification of these residues. And that's where we try to look for amino acids, or if there is any form, really any kind of difference between our control target that doesn't get impacted and the main target that has experienced the shock. Another thing that I have been interested in, and kind of links to all this, is trying to find the peak temperature achieved on impact. So like I said before, the conditions under which these occur are so important, because there is such a variation between observations, computational simulations, experiments. There's a significant difference between all of them. So we're trying to find a way where we can start linking things and try to understand, not only to say that, oh, this exists, but to understand why. Like, oh yes, it would exist because it achieved this energy, or it achieved this pressure. And one of the things that I'm looking at is trying to determine the temperature from the impact itself. So during hypervelocity impacts, you get a light flash when the projectile hits a target above certain speeds. On argon, it's above about 5 kilometers per second with the materials that we use. So we set up a high-speed camera to record this impact flash, which is technically called the self-luminous plume, but we call it impact flash more colloquially, but that's its real name. And here on the right-hand side, you can see a picture of it at the top. And at the bottom is the frame immediately after, where you can see all the ejecta shooting out of the ice target and the trailing light remaining. And that target was just a saltwater ice target that was hit with a small aluminium sphere that was 3 millimeters in diameter, but it was hit at 6 kilometers a second. So it can do, even though it's 3 millimeters in diameter, it can still do a surprising amount of damage. People don't really realize. So if I move on to the next slide. So what do we do with that? How do we actually get this temperature? How do we try to constrain these conditions in order to then to try and apply them to what we observe in space, or what we can get from information in space? So right here in the middle, I have a spectrum from literally that exact shot. So the shot I showed you above, which is saltwater ice at about 6 kilometers a second with an aluminium projectile. This is the spectrum that was produced. I know people don't really like graphs, but it wouldn't be a science talk without a graph. I'm sorry. You have to. It's the only thing I have to look forward to, showing people data. So what's quite interesting about this is, first of all, you can immediately identify what was in the target. So you get the aluminium lines and the aluminium oxide molecular lines, and these all directly relate to your projectile, which is very interesting because it means that you could potentially be using this technique to identify what hit your projectile, your impactor, whether this is aluminium or it could be something else. But it's very interesting that you can see that. And then you have the 2 sodium lines, and these are from the sodium chloride, the salt in our saltwater ice mixture. And the way we determine the temperature based on just this information alone is using the orange lines, which is the sodium lines, as you can see, using their relative intensity, so using their proportion to each other, and performing some quick maths, Boltzmann, I say quick, it's not that quick, Boltzmann distribution calculation, you can get approximate temperatures that were achieved on impact. However, over the last couple of years, I've been working with some collaborators at the University of Dundee. I've listed it here at the bottom, where we can actually do a lot of this, instead of doing a lot of this manually, we can now do it automatically. So they've developed a Python package called StarMelt. And essentially what that does is it can identify those lines, those emission lines automatically for you. And it can do all the maths for you. It can extract them and do that and just give you a final answer, which means that we can do it our way, the analog way, but then we can check it with this automated system. And that's interesting because this was originally used, made to look at things in the interstellar space. So actually being able to take something like that and use it within our solar system, which is much smaller confines, is quite interesting and very useful to show you again the collaboration that just happens between different departments in different places in the world, and how we can always help each other and borrow different tools to explain and use them for what we need. So what does this information actually tell us? This tells us that we can obtain the temperature for a certain mixture of target and projectile. We can determine the temperature that can achieve on impact. So if we create something, let's say when we make, hopefully, if we make cysteine, we can not only say that, oh, we have successfully made cysteine out of these products, we can also tell you that the temperature at which it happened at was this. So it's not just an issue of just telling you what you found, but how you found it, at what temperatures you found it. Because people right now all over the world are doing impacts onto things to try and see whether that could be the answer to the origin of life or the evolution of life in our solar system. But then again, if you don't have the conditions to compare it to, we're all a little bit blind, because what you do and might work for you might not work for me. A slight difference in composition of the ice target could have huge implications to what you get. So establishing the conditions is actually quite important, and will also help when we look back at these icy moons and we look at data we can obtain from them. If we do or don't find certain things, we can explain that. We can try and explain that if we know the conditions. So in summary, to sort of round up for you, the icy moons are becoming of increasing interest due to the presence of the subsurface oceans and the ingredients necessary for life. On the right-hand side here as well, sorry, I always found a really cute picture, is Jupiter, and there's little Europa, and you can see how tiny it is in comparison to the gas giant. But yet, despite being so tiny, it's so very interesting to us. And I think that's fascinating to think about, how a small body that was considered sterile up until however many years ago is now one of the most likely places to find life beyond Earth, as well as Enceladus, as well as Callisto, and a lot of the other icy moons that I didn't go into today. But yeah, so they are some of the most likely places to find life beyond Earth, primarily, like I said, due to the subsurface oceans and presence of hydrothermal vents, showing you that if on Earth we have life at 121 degrees Celsius at the bottom of the ocean, what's to say that that doesn't exist there? We would have never thought that was possible if we hadn't observed it here. So it really does beg the question. And then we have found that impacts could have definitely contributed to the presence of amino acids in our solar system. And therefore, that could be a part of understanding the origins of life. Because like I said, if we can make amino acids out of ingredients that already exist, and then we can start making them more complicated, and then get tertiary structures and 3D structures and protein and DNA, all of these are each step to creating more complete and more elaborate structures, organic structures, and life as we know it here. So will we really know if there is life on the icy moons of our outer solar system? Well, I'm glad you asked. Hopefully over the next few years, we're going to find out a lot more things. So as a lot of you probably already know, just at the end of last year on Christmas Day, the James Webb Space Telescope was launched. Here in the middle is a picture, and the James Webb Space Telescope has many objectives. It's going to shed light into the cosmic origins, exploring very, very old galaxies in our universe. But it's also going to probe whether the ingredients necessary for life are present in our outer solar system and beyond, and then looking at exoplanets. So we're very happy that this has finally been launched, and we can start getting data, actual observational data that's a lot more detailed, a lot more recent, and seeing how that goes. Then over the next few years, a lot more coming up literally in the next 2 to 3 years. So there's the JUICE mission, the Jupiter Icy Moon Explorer, and that's by the European Space Agency. And what they want to do is try to characterize the conditions that may have led to the emergence of habitable environments on the Jovian moons. But it's going to have a specific focus on ocean-bearing worlds, such as Ganymede, that I mentioned before, we didn't really go into, Europa, of course, and Callisto, that we also didn't really go into. And then we also have the Europa Clipper that's currently in the progress of being developed, where they intend to, it's going to be another flyby mission, and is going to try and explore Europa in a bit more detail, investigate its habitability further, and try to help in finding a site for the Europa lander, because the discussion, the idea is that we can actually send something there, like we did on Mars with the Mars rover, to send something there and either scoop something up from there or drill down. They still, I don't think they've sort of like finalized things, but they know that they want to do something similar to what we have done on Mars, where we can actually start obtaining samples from the ground and probe the habitability and probe these questions further and see if we can find biomarkers. Can we identify these biomarkers? Similarly, with Enceladus, there are discussions about flyby missions that could go through the plumes of Enceladus and try to collect some of that material. I was actually part of a team led by the University of Berkeley in California, and I was the person here at Kent to help them out, where we performed ice shots. So we made ice projectiles and we fired them onto a selection of targets because they were developing a detector system that they could put on one of these instruments that would fly through the plumes of Enceladus to try and collect information from those ice particles. So have those ice particles impact the substrate, but without it damaging anything that it has inside. So essentially we put a little bit of organic material inside those ice targets. And well, first we started with just organic targets, and then we put just ice, and then we put ice and organics. But there's a lot of steps to it, as with all experiments. But the point being that we essentially try to establish the best material to use if you want to capture those ice particles without damaging the organic material that could be embedded in them. And we have a lot of that work published. So if anyone is interested, I can always refer you to that. But yeah, so these are all things that are being discussed at the moment. And we are very much looking forward to all this information coming along. But in reality, we probably won't really know the answer until we have some kind of sample return mission, because observations are great and they can give you a very good idea of what's in there. But until you have your hands on a sample, an experimentalist like me, that's what we want. We want the real thing. I know spectroscopists love their spectra and their data, but it's not the same, because it's actually being able to find biomarkers is going to be hugely significant in and of itself. But once these missions establish these things further and we can hopefully get some return missions, that's when we'll know for sure. But hopefully over the next few years, with James Webb obviously having just launched and JUICE launching next year, we should have a whole amount of information coming in with respect to that, to finally answer the question as to whether there is life. But in reality, we might not even know if we don't go near down the hydrothermal vents. That's all a bit of a debate. We don't, at what point we may or may not see something is still being discussed. So yeah, just some quick acknowledgements quickly. So obviously I would always thank my collaborators and their institutions for always being willing to link projects together and try to come up with wacky ideas to try and make these questions a bit more approachable, and try to take the concepts that we see and try to replicate them in the lab and try to make sense of what we find or don't find, because what we don't find is just as important as what we do find in science. And then STFC, who are currently funding me, and of course the Royal Museums of Greenwich for having me today